Right, immunology recap. We have a phagocyte. We have a bacteria. Could be a virus, it's got antigen on its surface, non-self. You have a process phagocytosis, ingestion, digestion, destruction of your pathogen there. Okay? That in turn will lead to formation of our good friend, an APC, antigen presenting cell. Now, this in turn will stimulate an immune response. Two things we can look at with this. If we were to carry this on, we could have an attachment here of our friend, a B cell. We could have an attachment of a T cell, and also a T helper cell. Okay, nice and straightforward. So the idea is that these things will bind. If you remember going backwards, that each one of these is complementary to that particular antigen. If you remember clonal selection, you are generating B cells, T cells, and T helper cells, all with the same receptor proteins to a given antigen. You have 10 million of these when you're born, and you're constantly generating new ones at random. If one of these is complementary to that given antigen on a pathogen, you'll then bring about the next phase, our next response. So at this point, if we were to look at our friend, the humoral response, which is all involving antibodies, nice and easy. So humoral is all about antibodies. The idea of this B cell here will become activated because of our friend the T helper cell. If you remember, T helper cells will release cytokines. It's a molecule, a chemical, which basically fully activates this B cell. At that point, this B cell here, I'll pop it there looking like that, it will dissociate, it will then go off and undergo clonal expansion. Okay, so at that point, in effect, you've got mitosis. And what will happen that this B, activated B cell, will become, in effect, I can't fit it on there, but that will become a plasma cell. In addition to that plasma cell, you'll also get some differentiation, and you will form our good friends, the B memory cell. Okay, so your B memory cells will be long term, and the idea is we'll come back to that memory cell aspect going forward. Now these plasma cells will divide rapidly, producing lots of antibodies. Okay, those antibodies again will be in effect complementary to this original antigen on your pathogen. And if you remember the three things, it will lead to agglutination of pathogens, precipitation of toxins. So again, some bacteria release toxins, so it will precipitate those out, insoluble, and therefore they can be consumed by phagocytes. And your last one was opsonization, which in effect makes them more susceptible to the actions of phagocytes and macrophages. That collectively is your humoral response in the humours. If you remember this process forming plasma cells, these will divide rapidly, producing lots and lots of antibodies. And that floods the fluids of the body, blood plasma, blood tissues, etc. The B memory cells, those will reside for a long time afterwards, and it's these which basically confer this idea of immunity, presence of memory cells. So if this B memory cell comes across this particular pathogen again, this antigen, it will in turn activate in a secondary response and produce more plasma cells. Okay, so activation of the memory cell doesn't require a T helper cell for next time. Okay, so non-self phagocyte ingested, APC produced, complementary B cell, T helper cell activates, forms plasma cells produce antibodies, three means of action, typical 6 marker. How How does an infection with a bacteria bring about an immune response? You talk about phagocyte engulfing, APC, this idea of clonal selection, that you've got millions of different B cells, T cells and T helpers, and again, one will respond to that particular antigen. Differentiation activation of B cells to form plasma cells and also B memory cells, and the idea that produce antibodies leading to these, and that will give you realistically your 6 marks. Key terms there will be your clonal expansion, humoral, antibodies, all the buzzwords in there. If we look on this next side, we'll do it all in one hit. If we were to assume we have an infection, for example, from a virus or a cancer cell, they're the obvious ones to look at. Basically, you end up with a cell with non-self antigens on the surface. Very simply, we've got a cell that's infected. This will in effect give us our cell mediated response. Okay, so cell mediated, we're going to basically destroy infected cells. This will work in the fluids and around, so pathogens, bacteria, free floating viruses. This is once the viruses get inside cells, or if they've got cancer cells which start to mutate and therefore produce non-self antigens. And the idea is we will have a cell, so let's have an infected cell there. On the surface are some antigens from the virus. Okay, one of these T will bind to it, will also have the same sort of T helper. Again, this idea of cytokines. If we released, cytokines will activate this T cell. So the T cell divides to form, and we've mentioned last time, B cells become plasma cells. These T cells will become cytotoxic T cells. Yeah? We'll put a TC there for cytotoxic. And basically what these will do is will divide rapidly, attach to the host cell, the infected cell, and what they'll do, they'll release molecules, these perforins, molecules a bit like a life belt or a polo mint, which will basically bind to the membrane and allow the contents to leave via, in effect, osmotic lysis. Okay? It allows the contents of an infected cell to basically leak out of the cell, basically destroying infected cells. So the cell-mediated response will kill damaged or infected cells. As well as forming these cytotoxic, we're also going to have our T memory cells. So as before, B memory cells and T memory cells. And again, this leads to this idea, this concept of immunity. You're only immune to something if you've got memory cells to that particular thing. Again, these will reside in the system for pretty much a lifetime. Same sort of thing, if this comes across a given antigen, and again, every T memory cell is complementary to a given antigen, straight away it will differentiate, form it more cytotoxic, and bring about the death of all these infected cells. Okay? So just like the B memory cells, we don't need T helper cells to activate the memory cells. Okay? Something else that will also do, these cytokines will also speed up division and development of phagocytes and macrophages. So the cytokines will activate B and T cells to become plasma cells and cytotoxic T. The cytokines will also encourage phagocytes to replicate at the site of infection. So we're going to get more of those phagocytes in there, leading to more of this kind of stuff. I think that's everything we needed. Same sort of thing, the 6 marker. How does infection with a virus lead to an immune response? Complement T cell through clonal selection, activation by T helper cell, releasing cytokines. T cell differentiates to form a cytotoxic T cell or a killer T cell. I'd avoid the word killer, go for cytotoxic. That in turn attaches to infected cells, cells with viruses in, introduces these porphyrins, however you want to pronounce it, which leads ultimately to osmotic or cell lysis, osmotic lysis. It causes the contents of the cell to leak out, kills the cell, and therefore viruses can't be replicated. Memory cells will reside for a long time. Next time through, that's your response. I think that's all I wanted to mention on that one. Good stuff.